Okay, we still have a couple of minutes before we start. Okay, I guess we can start now. Though some people are still joining. So welcome everybody to the Protein Folding and Dynamics webinar. Unfortunately, this lecture was supposed to be originally by Jose Onuchik, but he could not join us today, and his lecture was postponed to April. But instead, we are very lucky to have a terrific lecturer today, Dr. Hagen Hofmann from the Weizmann Institute of Science. I just want to mention that the next lecture will be in 1 week from now. Because of the Biophysical Society meeting, we moved this lecture. So the next lecture is not in 2 weeks, but in 1 week. And it's going to be given by Nozomi Ando from Cornell University. But let's focus on today's lecture. So as I said, Dr. Hagen Hofmann, our own Hagen, agreed to give a lecture today. And I'm sure that's going to be a terrific lecture. Let me tell you a little bit about Hagen. Hagen did his PhD at the University of Halle in Germany, and then he postdoc with Ben Schuller in the University of Zurich. He did some really beautiful work during his postdoc there. I will just mention 2 pieces of work. One is the study of the conformations of a protein sequestered within the internal space of the molecular chaperone GRO-EL. And another seminal work is the measurement of the scaling laws of unfolded proteins under native conditions. In 2014, Hagen joined the Weizmann Institute as an assistant professor, where he's ever since. And Hagen's work revolves around a new field that is starting, which he calls molecular systems biophysics, meaning looking at proteins at the molecular level, trying to understand their properties, but in the context of other molecules, in the context of even networks of molecules. And Hagen came up with some beautiful ideas for new measurements, new work over the years. And I must say that one of the most important, most exciting features about his work is the rigorous insertion of models and theoretical concepts from polymer physics and from chemical physics into his work on biological systems. Today, Hagen is going to tell us about allostery in DNA and how it drives phenotype switching. So Hagen, the floor is yours now. I will stop sharing and you can share your screen.
Thank you very much, Gilad.
Let me just mention, Hagen, before you start, just one more word. We are going to have a question and answer session in the end. Please write your questions in the chat, and then I will call you one by one to unmute yourself and ask the question. But during the talk, please keep your microphone muted. Thank you.
Okay. Thank you very much, Gilad, for the very kind introduction, and particularly thanks Ben and Gilad for inviting me to jump in for Jose. It's really a pity that he cannot give a talk today about energy landscapes and chromosomes. But as promised on Twitter and also on our website, Jose will for sure give a talk on April 19. So today it's going to be about allostery, and particularly on allostery in DNA. And actually, I would like to start this talk with two terms that are probably familiar to you, which are chance and necessity. So these are two terms that you probably know from the title of a book written by Jacques Monod many decades ago. And what Jacques Monod tried to do in this book is to make a synthesis and to reconcile Darwinian evolution on one hand side with what was at that time known as modern molecular biology. And what he means with this term necessity is basically the sort of necessity with which biological processes seemingly occur. If you think about enzymes or molecular machines that work with a certain, nowadays we would probably call it a mechanism. And the term chance, of course, refers to mutations that are accumulated over the course of evolution. However, it turns out that chance, stochasticity, or randomness, if you wish, have a way more immediate impact on biological systems. And this is illustrated in this statistics here, which comes from the lab of Sunay Shi at Harvard. And what they did in this experiment was to count the number of proteins of different types in an E. coli cell. And you see that these molecule numbers really go from thousands of molecules per cell down to less than one. And to me, one of the surprising findings is that there are so many molecules, proteins found in an E. coli cell that are really present at extremely low copy number, down to one molecule per cell and even less. And in this respect, it turns out that transcription factors are not really an exception. So very often, transcription factors can also be found at such low concentrations. And if this happens, you can imagine that gene expression itself becomes a stochastic process, because it simply takes a stochastic amount of time until a single copy of a transcription factor can find its target in the genome. And the result of this stochastic gene expression, or gene expression noise, if you wish, is illustrated in this example here. So this is an experiment done by Michael Elowitz at Caltech also many years ago. What you see here is a population of bacterial cells. These are Bacillus ceteris cells, and they are all descendants of one and the same cell, which means all these cells are basically genetically identical, but nevertheless, despite the fact that they all share the same genome, they already show quite disparate phenotypes. You see these black cells here, which are vegetative cells, meaning normal bacterial cells. Then you find these white blobs, which are called spores. And finally, you also find a third phenotype, which is this magenta cell here that doesn't really divide, but just elongates, which is called a competent cell. Now, what actually happens in this case is that these bacteria are starving. And if they are starving, they have a rather simple and very effective survival strategy, which is to form these spores, which means they squeeze out all water and lower their metabolic rate. And in this way become extremely long-lived but dormant entities. And NASA reported that they can actually survive 3 weeks in outer space. Now, it turns out that when these vegetative cells develop into spores, actually some of the cells die along the way. And if they die, they also expel their genetic material into the solution. And that's why there's a third phenotype, which is called the competent phenotype. And in this phenotype, the cells express a membrane machinery, essentially a DNA import machinery that can bind the DNA in the extracellular space, transports it into the cell, and then integrates bits and pieces of the DNA into their own genome. And so essentially these competent cells are rescuer cells that take care of this lost genetic information. Now, if everything in biology would be deterministic, then Bacillus subtilis would either form spores or it would form competent cells. But it turns out that the competent cells only form in 5% of all cases. And it's actually unpredictable which of those vegetative cells will eventually form this competent phenotype. And that implies that the actual molecular trigger for forming the competent phenotype is a stochastic event. And the question that we had here was to understand what's the molecular basis of this stochastic trigger. And so everything, again, starts with a transcription factor. The transcription factor in this particular case is called COMK. It's a rather small protein, just 22 kilodalton in size. And when we started working on this project, there was not a lot of information about COMK. It was clear it has no homologs in any other organism, which doesn't make it a particularly attractive target, I have to admit. And on top of that, it was also and still is very aggregation prone. So it aggregates down to concentrations of about a micromolar. And so since there was naturally also no structural information due to this aggregation tendency, one of our first questions was that maybe COMK is partially disordered and therefore very unstable, and that may explain its strong aggregation tendency. And so the first thing we did was to just run a ponder prediction to see whether we have significant amounts of disorder in this protein. And it actually turns out that there are some regions of COMK that are disordered, but these stretches do not really extend beyond 20 amino acids. So a big part of the protein is actually predicted to be well folded. Now, it took some time until we managed to determine the thermodynamic stability precisely for COMK. And when we did that, it turned out that it was in fact really low. So just 8 kilojoule per mole. If you do the math, that means that at any given moment, about 3% of this protein is totally unfolded if you would believe the two-state approximation for such a unstable protein. So in summary, COMK is a very unstable transcription factor, but luckily the functional aspects of COMK had been studied well over the past decades, mainly by microbiologists and systems biologists. And as mentioned, COMK acts as a transcription factor. It binds to a promoter that is sitting in front of a large operon that contains all the genes that code for this DNA import machinery. And simultaneously, to activate this, this expression of the DNA import machinery, COMK can also bind to the promoter in front of its own gene and can self-amplify. And so now the third ingredient you need to know is that, of course, COMK is unstable and can also be rapidly degraded. And so if you now combine these factors, the current model of how this stochastic phenotype switch happens is the following. Average levels of COMK and these bacillus cells are extremely low, due to very quick degradation, which means COMK can only sporadically bind to the promoter in front of its gene and accumulate a little bit, but then gets degraded quickly, which leads to these stochastic fluctuations in its copy number. But since these fluctuations are stochastic, they can also by chance reach rather high values. And the idea is now that once these fluctuations exceed a critical threshold, then for a certain mechanism and reason that still needs to be specified, COMK can stably bind to its promoter. It can self-amplify in the cell to high numbers, and then it can actually start the expression of the DNA import machinery in cells become competent. So that's really a beautiful mechanism that was unraveled by microbiologists and systems biologists. And for us, the big question is, where's the threshold coming from? So how can you actually build a threshold at a molecular level? And to us, this threshold sort of reminded us a little bit of these electronic analog to digital converters. And like in those analog to digital converters, also in this biological example of COMK in stochastic phenotype switching, you basically need a stepwise or step-like response function. Just that in our case, the response function is actually the binding curve of COMK to its actual promoter. And so if you have such a highly sigmoidal binding curve, then you indeed have a regime at which changes in COMK concentration don't really matter. The promoter will remain inactive, but as soon as COMK crosses a rather narrow range in concentrations, the promoter is switched on in a sort of all or none manner. Now, I think we all know that these sigmoidal binding curves basically imply and require cooperativity in binding. And so that means that at least we have to have two COMK molecules that bind to the promoter. And on top of that, they also need to bind in a cooperative manner. Now, cooperativity empirically is very often quantified using these rather generic Hill functions, where you have the concentration of your ligand up to a certain exponent, which is called the Hill exponent or Hill coefficient. And what this number tells you is basically the minimum number of ligands that can bind to your receptor. So the whole question of where is the threshold coming from basically cooks down to the question of, can COMK bind in a cooperative manner to its promoter? And if yes, how is it doing that? And so in order to understand that, we first had a look at the promoter for COMK. And it turns out the promoter has two boxes where COMK can bind that had been understood and investigated using DNA footprinting experiments many years ago. And these two binding boxes are separated by a spacer. And now, depending on the gene you look at, you find spacers of 8, 18, up to 31 base pair length for this promoter. Now, inside such a binding box, you find a quite interesting sequence, namely 4 adenines, 5 random nucleotides, followed by 4 Ts. And so it has been shown in the past 20 years that these so-called stretches of A's, or also A tracts, can actually bend DNA towards the minor groove, such that the minor groove gets narrower in those A tract sequences. And so the first question that we wanted to address was to understand whether the topology of this promoter is actually straight B DNA or is curved DNA. And so in all the following slides, I will show you the schematics for this promoter. So we have here box 1, box 2, and the 2 stretches, the A stretch and the T stretch. And in order to understand the topology of this promoter, we actually use single molecule FET. And we produced many constructs of this promoter with a donor and an acceptor dye. If we excite the donor, it can either emit a photon or it can transfer the energy to the acceptor, and then the acceptor emits a photon. And so this transfer of energy is depending on the distance between the two. I think most of you will be familiar with the FRET technology, so I don't have to go into too many details here. And so we did all these experiments in vitro. So we had these DNA promoter constructs with the 2 dyes. These molecules were diffusing randomly through the confocal spot of our microscope. And each time we had a molecule on the spot, we get this burst of photons, in red for the acceptor and green for the donor. And by just dividing the number of acceptor photons by the total number of photons, we actually get the thread efficiency, of course, we have to do all kinds of corrections for quantum yields, detector efficiencies, leakage, and so on and so forth. I don't really want to bore you with all those details. The important fact to remember is that the thread efficiency is actually inversely proportional to the distance between the two dyes. And so by counting enough of those molecules, we can construct these thread histograms. And these thread histograms give us one type of important information, and this is the position. Because this position is actually related to the average distance between the two fluorophores. Now, the width in these histograms is not really of high information content, because in these particular cases, the width is basically determined by shot noise, which means by the uncertainty in determining thread that we have due to the fact that we can only count and monitor and probe a limited number of photons for each molecule. Okay, so once we have these average thread efficiencies and we know what we are looking for, we can actually make a little prediction. We have a pretty good idea of how bDNA should look like. And so that means we can simply pre-calculate the profile of thread efficiencies that we would expect for bDNA. And if we do this experiment that I just described at low pH values, we actually find a pretty good agreement between the measured values and the predicted values. Now, at low pH values, the phosphates in the DNA are protonated, and so the flexibility of the DNA is much greater, which means it has a lower persistence length. And it has actually been shown in previous experiments that the capability of bending DNA by ATRACs is lost at lower persistence lengths. And so what we would expect, if there is some curvature, is that if we go up with the pH to neutral pH, that we find some deviations. And indeed, this is the case. So at neutral pH, we indeed find that thread efficiencies are significantly higher than what we would expect for simple bDNA, implying that indeed the promoter has not a bDNA structure, but is in a curved or bent state. Now, of course, now the question is it rigidly bent, or do we have fluctuations between bent and unbent states? We can answer that question by going to extremely fast timescales. For example, we can monitor and measure the fluorescence lifetime of our dyes. And this fluorescence lifetime of our dyes is expected to have a very clear dependence on the thread efficiency. And so first, the theory predicts that the fluorescence lifetime of the donor should scale linearly with the thread efficiency if you have a static distance. And that means if indeed we have a rigid band, all our constructs that we measured should come to lie close to this dashed line, which indeed happens for most of the constructs. However, there are also some slight deviations. And if we fit the distance distribution to all those data, we find that actually if there is a distribution, then it has a width of about 0.7 nanometers. And we can confirm this using a slightly different methodology. We can just take one of those constructs and fit directly the fluorescence decays of the donor and of the acceptor, and that gives nearly the same width. Now, the question is this a huge heterogeneity that can come from differences in bending, or is this actually negligible? Well, it's for sure not negligible. But it turns out that we have to attach our dyes with rather long linkers to the DNA. And the effect of those linkers has been estimated in previous experiments to be about 0.6 nanometer. And so that really coincides pretty well with what we measure. And so the bottom line of all of those experiments is that, yes, there's a distance distribution, but this distribution comes from the dye linkers. And apart from that, the DNA is really rigidly bent, at least at the length scales of a few nanometer that we can probe with thread. Now, after having understood that indeed the promoter is curved, the next question is, can we probe binding? And of course we can. We simply use the same method, single molecule thread. And in this particular case, we now label one box. So we place a donor dye at the beginning of a box and an acceptor dye at the end of a box. And then we simply add our transcription factor, comp-K. And what we find is that with increasing comp-K concentrations, the thread efficiency distribution shifts actually to lower values. And I have to admit, the shift is not very big. It's a rather tiny shift. And if we have to put some numbers on, it would be a distance change of about 3 angstrom. And that means the two fluorophores, the distance between the two fluorophores increases by about 3 angstrom, which is not really a lot. However, it's just enough to deconvolute all those distributions in the middle here by the distribution of the totally saturated promoter and the totally unsaturated promoter. And that actually can be used to calculate and construct a binding curve. And in fact, this binding curve really shows high cooperativity in binding COMK with a Hill exponent of 3.6. Now, the same experiment can, of course, be repeated. We have 2 boxes. Here we had the dice on box 1. We now put the dice on box 2. We repeat the experiment. We get exactly the same result, a Hill coefficient of 3.4. Now, what this number means, and I mentioned it in the beginning, is that at least we have 4 COMK molecules that actually bind to one of those boxes. And in principle, this could have been the end of the story, but we wanted, because it answers the question of whether COMK binds cooperatively or not, but we wanted to do one control before we stopped. And the control was rather simple. So instead of monitoring binding to the full-length promoter, we monitor binding to the same box, but in the absence of the first box. And when we did that, it came as really a surprise when we found that the cooperativity dropped down by about a factor of 2. And we find the same drop in cooperativity when we did the reverse experiment. So we labeled box 1 in the absence of the second box. Again, we find a factor of 2 drop in the cooperativity. Now, this Hill exponent implies that there are just about 2 molecules of COMK binding to one of those boxes. And that actually leaves us with a little conundrum. So if we have 2 molecules binding to one box, then we still are left with an explanation, with the need to explain how we find 4 molecules binding to the promoter by probing one box in the presence of the other. And that, of course, requires cooperativity. It requires a communication between the 2 sides through space somehow. And I would like to mention that the binding sites are really far away from each other. So we're not talking about a few angstroms. We're talking about 6 to 10 nanometers, depending on the different promoters we're looking at. Now, our first idea of how this communication may happen was sort of the classical textbook idea of looping the DNA, because in that way you can allow communication of proteins on one side with proteins on the other box. And we thought that this may be a good idea because there's ample indication on other studies that proteins can really bend strongly DNA. One very famous example is shown here on the right side. That's integration host factor bound to DNA that really bends DNA by nearly 180 degrees. Yet there is an interesting twist to the structure here, because in order to obtain this structure, the authors had to nick one strand of the DNA. And this nicking of one strand of the DNA made the complex way more stable and allowed the crystallization of this protein DNA complex. And we thought maybe that's also a good way of proving whether our looping model is correct. So if we would nick one strand of the DNA, looping should get easier, and that in turn should maybe result in even higher Hill exponents. And so that experiment was easy enough to be tried. And so what I do here is I show you exactly the same data that I showed you before, just now in a log-log plot, which is also called a Hill plot. And the advantage of a Hill plot is simply that you can directly read off the Hill exponent as slope from the data. And so here we can have our data for the 2-box promoter, first box labeled Hill exponent 3.6. Here we have the data of the single box promoter, and our prediction was now that by nicking the DNA, we would get an even steeper slope. Unfortunately, the opposite was true. So when we did nick the DNA, the Hill exponent again dropped by about a factor of 2 and nearly reached the slope that we find also for our single box construct. And so at that point, we really came to that end because the experiment basically disproved our hypothesis, but didn't give us a clue of what an alternative mechanism for communication between the two boxes could be. And so we definitely wanted to get structural information about this protein DNA complex, but certainly X-ray crystallography and NMR spectroscopy were out of discussion due to the solubility issues we had. And we were basically left with cryo-electron microscopy. But also there, we had some challenges because the DNA itself is just 50 kilodalton, and plus 4 COM-K molecules gives you roughly 100 kilodalton of a complex where we didn't even know whether it's rigid enough to be seen in cryo-electron microscopy. But we gave it a try anyway, and it turned out that when we just picked enough particles and class averaged them, we indeed found that the particles were roughly dividing into two major classes. One class that was simply blank DNA, and a second class where we had extra electron density at positions that really sort of roughly were at the positions where we expect COM-K to bind. And by doing more experiments also with tilted angle, we actually could also overcome severe orientational preferences that we had in those data sets and generate a three-dimensional cryo-EM map of the protein in complex with the promoter. And so that's what you see here. So in gray, the DNA, in red, the COM-K molecules. And so actually, they are really sitting nicely at the positions where we would expect them to reside based on the old DNA footprinting experiments. But on top of that, we also find an interesting C2 symmetry in one of those boxes. And C2 symmetry sort of is really indicative of having two molecules or two entities bound to one box. And indeed, when we do volume calculations of this density, it turns out to fit quite well to a stoichiometry of 2 COM-K molecules per one box, in total 4 COM-K molecules per promoter. When we turn the structure on the side, we find that COM-K only binds to one side of the DNA, and this is explained by simple arithmetics. So from the middle of one binding box to the middle of the other binding box, it's nearly precisely 3 turns, which means the binding sites for COM-K face actually the same side of the cylinder. And we can do the same experiment with another promoter here for the 8 base pair promoter. And there we only have 2 turns between the two sides. And again, the molecules, COM-K is sitting on the same side of the DNA cylinder. Now, what these experiments told us is that definitely there is no looping. But when I presented earlier results on a conference, I got the remark that, well, during plunge freezing that you do in cryo-electron microscopy, you may actually destabilize looped complexes, and what you're left with are these straight protein DNA complexes. And we were looking for a way of understanding whether this is actually the case here. And so that required us to directly compare our cryo-EM data with our single molecule FRET data. And there are actually 2 ways of doing that. And the first way is to try to compute Hill exponents directly from the cryo-EM data. And so we can do this by going back to the raw data and actually counting how many particles do we find where we have just blank DNA, how many particles do we find with 1, 2, 3, 4 molecules of COM-K bound. So if we get this binding statistics, we can actually compare it to the statistics we would expect for independent binding of COM-K molecules, which would simply be a binomial distribution. And so by doing this comparison, we found that cryo-EM, based on the cryo-EM results, we would expect a Hill exponent of 4. In solution with FRET, we measure 3.6. The same for the 8 base pair promoter, because of 3.9 from cryo-EM, about 4 from single molecule FRET. And for the long spacer DNA, we find 2.6 versus 2.3. So in overall, in very good agreement. And so a second way of confirming that FRET and cryo-EM is in good accord is, of course, to model DNA into our cryo-EM density, attach the dyes the donor, the acceptor for the two boxes, and then to calculate the thread efficiency and compare it with what we measure. And actually, when we do this, we actually find pretty good agreement between the calculated values and the values that we actually measure. So overall, single molecule thread seems to be in good agreement with what we get from cryo-EM, but still this doesn't resolve the conundrum of how the communication between proteins that bind to this side happens with proteins that bind to that side, because there's definitely no protein density in between here. Now, a clue of how this communication might happen comes when we analyzed the topology of the DNA in those complexes. And it turned out that the topology in the free DNA, in the absence of any protein, showed much higher bending angles than what we found in the complex. And so this now gave us an idea of how the communication may actually take place. And in particular, when we overlaid the EM density of COMK with the atomic model, we saw that COMK is really facing the minor groove of the DNA. And remember, in the beginning, I told you the minor groove in those 8-track sequences is way more narrow than in bDNA. And so that implies a mechanism in which we start with a curve promoter, binding of COMK to one side, actually widens the minor groove, that introduces tension that propagates along the DNA and actually leads to a widening of the minor groove on the other side, on the other box. And in fact, if we sort of do a little PCA of our protein complexes, we see that there is actually quite some variability. So what you see here is a morph of the first 3 components in the PCA that account for more than 90% of all the variability. And such a bending mechanism or a tension propagation mechanism could in fact be at work. However, we didn't really know from the physics-based perspective whether tension propagation is at all possible. But luckily for us, a sort of similar scenario had been studied in the past, also theoretically. And it has actually been shown that DNA under tension can actually mediate cooperativity between distant sites. And so what these authors, Rudnick and Brunsma, did was to use a worm-like chain model to calculate the energy that is required to introduce 2 kinks into DNA. And what they found is that if they reduce the distance between the 2 kinks, they found that it's becoming more and more harder to introduce the 2 kinks. So it requires more and more energy. Now, the theory that they used was a 2-dimensional theory. So there's also an alternative arrangement of those kinks, which is called the antisymmetric arrangement, where we have positive curvature here, negative curvature there. And for this particular case, it turned out it requires less and less energy to introduce the kinks the closer they are. And this can be intuitively understood because if you overlay one type of kink with positive curvature with another kink with negative curvature, you end up with a DNA that has no curvature at all. And so it doesn't require any energy to introduce kinks that are not present, basically. Now, the only problem in applying the theory is, or one of the problems applying the theory is, first of all, this applies for DNA that is under tension. And the second thing is that you cannot simply move binding sites along one side of the cylinder. If you move binding sites away from each other, you do this base pair-wise, and that means at the same time you also rotate along the long axis of the DNA, which actually would give us a damped oscillation as a prediction for the energy to introduce those 2 kinks. And so if this is the prediction for the energy required to introduce 2 kinks, of course, that would also be the prediction of what we would expect for the cooperativity, for the communication between the 2 sites. And so that was really an experiment that we could do. And so we designed different constructs of the promoter with different spacer lengths and determined Hill exponents for all of them. And what we found was indeed a damped oscillation in those Hill exponents. So we go here from really high Hill exponents of 4 down to almost 2 and then up again. And so the upper curve that you see here is basically for the symmetric case. The lower curve is for the antisymmetric case. And the periodicity that we find is an actually pretty good agreement with what we would expect for DNA. Okay, so far so good. However, these are Hill exponents, and what we really would like to understand is what is the coupling free energy between the two binding sites, or in other words, by how much is the affinity in the second box increased if we bind proteins to the first box? And in order to answer that question, we need a mechanistic binding model. And this is the model that we actually derived based on our cryo-EM experiments. So the first COMK monomer can bind to any of those 4 sites. The second one, however, binds preferentially to the site where there is already a protein molecule, COMK molecule sitting. And we assume that based on the really huge interaction surface between the 2 COMKs that we find in the cryo-EM data. And then, of course, we have here the DNA-mediated allostery. So the third COMK molecules binds with a factor J stronger compared to the first one. And so using this model, we fitted all our data again and determined this J factor. And I show it to you now in terms of free energy. So the minus logarithm of this J factor. And it turns out that these coupling free energies also oscillate. The scatter is way bigger than for the Hill exponents because the fits are more complicated to perform. There are more parameters to determine. However, the coupling free energies are as high as 6 kT, which is actually twice the stability of the protein, if you want to have something to compare to. And actually, 6 kT implies that there is a 400-fold increase in affinity in box 2 if we have proteins bound already to box 1. So that's a massive increase in affinity that is mediated actually through DNA and not via protein-protein interactions. Now, there are basically 3 important questions that are still left to answer, and unfortunately, I cannot answer all of them. The first one is, what is the actual mechanism of communication? So can we say something about this? Secondly, what are the sequence determinants? So if we change something in the spacer, would it change the cooperativity? And thirdly, what would be the speed of the propagation of this allosteric signal from this box to that box? And so what we can say so far is that the sequence of the spacer doesn't seem to matter too much. So here we again have the wild type promoter with a Hill exponent of 3.5. That's the limit that we find for one box, basically no cooperativity between the 2 boxes. And so if we, for example, increase the GC content and this way make the spacer way more stable, we do not find a huge effect. We find a slight drop in the Hill exponent, but nothing really massive. We also introduced a mismatch of 4 bases, but even this didn't kill the cooperativity. So it was about the same effect as rigidifying the spacer. Only if we introduce a nick in one of the 2 strands did we get down to the Hill exponent of 1 box. So that really did destroy the cooperativity. And so what this means is that it's actually the 2 backbones that are important in order to allow tension to build up between the 2 binding boxes for COMK. Now, a second question that we had was whether we can boost the affinity basically to much higher numbers by just adding more and more boxes. And because that could be a very simple strategy in nature to tune cooperativity and to get to really steep dose response curves or binding curves. But it turns out that this is not really the case. So we made constructs where we had 1 box, 2 boxes, 3 and 4 boxes. And it turns out that cooperativity is increasing up to 2 boxes. Afterwards, it's not increasing anymore. It's even slightly decreasing. And so this tells us that communication between boxes can only really happen between neighbored boxes and not across boxes. And we really sort of are not completely clear about the origin, the molecular origin of this nearest neighbor interaction, actually the decrease in the Hill exponent could even imply that binding molecules here and binding COMKs here may lead to conflicting structural signals on the middle box, and that could even lead to a decrease in the Hill exponent. So in other words, there may be even frustration if we put and place too many binding boxes in one promoter. Okay, so this brings me to almost the conclusion of my talk. So what I showed you is that binding of transcription factors to one side can massively increase the affinity on another side of the DNA, and this really across distances of many nanometers. So the 18 base pair promoter basically implies that it's 6 nanometer difference between the two boxes, but we even find cooperativity in promoters where we have 31 base pair spaces. And so if you think about this, this means that you can actually nicely tune the dose response curve, the binding curve for transcription factor, but just moving the two binding sites closer together or further apart from each other. And this, of course, may have some functional and some engineering relevance if you think about designing genetic circuits, because cooperativity in transcription factor binding is not only important for this particular phenotype switch, but it's important in general for the dynamics of genetic networks. And I just want to mention one particular example, which is a very famous example of a synthetic genetic circuit. It's called the repressilator. And what you have here is are basically 3 genes. Each gene codes for a repressor. The gene X codes for a repressor that represses the expression of Y represses Z, and so on and so forth. That's a synthetic system that had been studied by Michael Elowitz and Stan Leibler many years ago. And what they found is that this system actually leads to sustained oscillations in E. coli cells. And if you model this system and you really take a closer look, you will find that in order to obtain these sustained oscillations, you need cooperativity in binding these repressors to the DNA, to the promoter in front of the respective next gene. And so changing this cooperativity really changes the dynamics of the system massively. So that just shows that transcription factor binding cooperativity to DNA is a really important factor that dictates the dynamics of genetic circuits, that is important for stochastic phenotype switches. And we just gave one example of how this cooperativity can be achieved, not via designing proteins, but via designing DNA promoters. Okay, so this brings me now really to the final conclusion, which admittedly sounds a little bit dull, but effectively, this is really the take-home message. DNA is way more intelligent than we think, and it's not just a passive carrier of information. It can actually really have functional and mechanistic functions in transcription factor binding. And so, I don't want to finish without thanking all the people, particularly Gabi Rosenblum, who did all the single molecule experiments, and he tied up with Nadav Elat from the microscopy facility at Weizmann Institute, and they both did a great job in doing all these cryo-electron microscopy experiments. Chaim Rosenberg did an excellent job in fitting DNA models into cryo-EM densities. Jacob Jungwirth from Gilad Aran's lab actually helped in comparing our Fred values with the actual cryo-EM data, and also Felix Wiggers was involved in early stages of the project. And so, of course, I thank funding, and I thank you for your kind of attention. Thanks a lot.
Okay, thank you, Hagen, for this beautiful talk. And now we have time for questions. So the first question is from Felix Ritort. Felix, you can unmute yourself and ask the question.
Okay, thank you very much, Hagen. It was a really very interesting talk. I would like to better understand this mechanism of tension propagation that leads to this cooperativity effect. Is the driving force of this tension propagation the stacking of the adenines that are on one of the strands of the DNA? Is there a cumulative, let's say, a stacking on this poly A or whatever sequences that give you this bending and therefore give rise to this propagation effect? Would a random sequence do the same? I mean, could you?
No, a random sequence wouldn't do the same. So you definitely need a row of adenines in order to get a band structure. And so I don't think that the molecular reason for this deviation from bDNA structure is very easy to pinpoint. So it has to do with stacking interactions between neighbored AT base pairs that actually lead to a little buckling of the base pairs, and then to the slightly distorted geometry of the DNA. But it really depends on the details of the chemistry of the AT bases. This is basically all I can really say about that. I was actually also surprised to see that because ATGC, I mean, apart from one hydrogen bond, they look very similar, but apparently they are quite different.
Yeah, that poly A and poly G, I think, are different. I mean, I think poly A has an accumulated stacking, at least in single-stranded DNA. That's my understanding. But
right, but if you think about double-stranded DNA, and so you think about the GC base pair and an AT base pair, I mean, of course they're slightly different, but not sort of orders of magnitude different. So I would have expected they behave the same. But apparently these bending propensities of AT tracts, they're well known since many years, and it's also a field that is rather vivid.
Thank you, Hagen. Congratulations for the beautiful talk. Thank you.
Okay, the next question by David Nesbitt.
Yeah, Hagen, let me just add, that was really a stunning talk, a gorgeous data and really heroic analysis, you know, layer on layer on layer of beauty. I'm wondering,
so much.
You talked at the beginning about how the tethers, the flexible tethers for your dyes basically add and give you some type of an effective length blurring that was masking and maybe masking, but basically sort of influencing some of the analysis. I'm wondering if there are ways in which looking at the polarization statistics from your FRET pairs might be able to help additionally and maybe provide additional support or confirmation of these really gorgeous mechanisms.
Yeah, that's actually a fantastic suggestion, and you're absolutely right. So, but we have to distinguish two scenarios. So there is depolarization, certainly due to the rotation of the dye.
Correct.
And then we have a second effect, which is basically the positional variability due to the long linker. And so typically the two are on two different time regimes. The rotational dynamics of the dye is typically below a nanosecond, so in the orders of hundreds of picoseconds. Whereas the positional dynamics of the dye is on a longer time scale than FRET. And then the fluorescence lifetime. So this has been investigated in depth by Klaus Seidel and his lab about 12 years ago, where they showed that actually the correct averaging regime is the one in which you can safely assume the so-called kappa square being 2/3, because the dyes are really averaging,
really
rotating freely, but at the same time, the motion of the dye in space is actually on a slower time scale. And you're right. I mean, fluorescence polarization experiments or anisotropy decays would certainly help. We didn't do this in this case because this is a bit more tedious when you do confocal experiments, because you don't have the 90-degree geometry that you typically like to have. And so analyzing those decays, it's a bit more tricky. It's not complicated. You just need to have two additional correction factors, but we didn't do it in this case. But yes, you're absolutely correct. In principle, this motional dynamics should also be visible in the decays of the fluorescence anisotropy experiments.
Beautiful talk. Thank you again.
Yeah, thanks, David.
Okay, Tim Craggs.
Hi there, Hagen. Wonderful stuff. I'm intrigued as to what you think will happen if your DNA was under, say, slight negative supercoiling. Is that going to enhance this effect, or
Yes.
which I would imagine? But I'd love to just comment on what you thought would happen in that case.
Yes, yes. Actually, I cannot say anything based on our data, but I can say based on the theory that I roughly touched. Because in theory, so based on the swarm-like chain model, the communication between the two sides only works if you have the DNA being under tension. So in our experiments, the DNA is not under tension. So somehow the theory is not really absolutely adequate for what we do. But you can imagine that if you supercoil DNA, you really satisfy this particular case of having you create tension in the DNA, and then you should definitely have this communication. And this was actually one of the driving forces for people to think about long-range communication of transcription factor binding. Yeah, that's an excellent comment. Absolutely true.
Jörg Enderlein?
Yeah, thank you. Super talk, Hagen. But it was already my question I wanted to put here. So your model is for DNA under tension, and there was this F cosine theta term. So
Right.
in your case, the DNA is completely free, and I
Right.
guess also not supercoiled. So how could you explain then this interaction for a free worm-like chain? Any idea?
Well, the only idea that comes to my mind, so there are two possibilities. Either sort of protein binding, maybe either protein binding of the first two proteins sort of introduces tension, or the A-tract sequences actually already sort of introduce some tension in the intermediate sequence between the two boxes. But it's a very good question. Actually, we have no clue where this tension is coming from. It just turns out empirically that the theory actually sort of gives the right prediction, but maybe even for the wrong reason. So that could well be the case.
Yeah, thank you very much.
Yeah.
Super talk.
Yeah.
Ben has a question.
Yeah, so thank you also from my side, Hagen. Really spectacular. I'm very impressed.
Thanks, Ben.
I have a question regarding your hill plots. So you had also a dashed line in your hill plots. Was that your full model, including the coupling free energies?
Yes.
And my question was, what kinds of different variants did you try there? Is that, I mean, what you chose is a very reasonable and probably sort of the simplest model you could choose, but I'm sure you tried a few different things. I was wondering which sort of boundaries you can place on possible models that describe the system.
Yeah, that's a good question. Actually, the first models that we actually tried. Let me show this again. The first models we tried were actually based on a simple sort of propagation mechanism, very similar to alpha helix formation, right? So that you have the first thing binding here, then comes the second, then the third, and the fourth. That was one model. I'm pretty sure it could have worked and described the data. Because it's just a matter of the number of parameters. And this is a general problem with all of these induced fit type models compared to Monod-Wyman-Chanier models, which are a bit different. But in these Koshland-like induced fit models, you end up with products of parameters, as you see here, the J, the sigma, and in the S here is actually also the original K in. Which really makes it complicated to fit. And so what we did in order to obtain rigorous parameters, or at least parameters with a decent error, was to constrain the sigma, which means the protein-protein interaction parameter, to be the same for all the different constructs that we measured, because we didn't have any indication that for the 18 base pair or the 8 base pair promoter, the COM-K density looked very different. And so that was one key in order to get to fit the J out here. Well, apart from that, Actually, I don't remember how many models I've tried. I mean, that whole project went over 4 years, and I think the first models I tried after 1.5 years. But it's sort of, this turned out to be the most simple model I could have imagined, with just 3 parameters. But already these 3 parameters are complicated. I would have loved to have a model with 1 parameter. That would have been my goal.
Thank you, Hagen.
Okay, we have Nancy Ford now. Nancy, are you still there?
Yes, yeah. Sorry, I needed to unmute. Beautiful, beautiful work, Hagen.
Thanks, Nancy.
Amazing. I was just curious about trying to further modulate this allosteric coupling, and if it's through tension. If you nicked outside of the boxes, do you think that would allow for an increased coupling because you're now relieving that requirement to deform further down the DNA?
This is actually an interesting question. I don't know. We never tried. We didn't even think about it because we always thought in terms of what happens between the 2 boxes, because that's where the strain needs to build up. But you're actually right. I mean, it also builds up on the sides, but we never touched that issue. I can't really say, but my guess is that the cooperativity between the 2 sides is likely not going to be very different, because you're going to have free ends in either way. I mean, it may be a different situation if we would incorporate this part in a plasmid.
Yeah.
So, and maybe even include supercoiling. Of course, I'm pretty sure that would change things dramatically. But introducing a nick outside the boxes and the spacer, I don't think that would be sort of equivalent of just shortening and increasing the length of the 3' and 5' outside the boxes. And I think this is something that we actually have constructs where the outside lengths are a little bit different, not on purpose, but just happen to occur. Yeah, I don't think this would matter much.
Okay, thank you.
Mm-hmm.
Ryan Hayes.
Yeah, I had a question whether you think some of the tension that you're seeing might be coming from electrostatics. You mentioned changing pH and how that neutralizes the backbone, and that would reduce self-repulsion.
Mm-hmm.
Salt concentration can also have an effect on it. I was just curious if you thought about that as a possible source of the tension.
We never. That's a very good. It's a very good point. We never checked it, but it's true that already addition of salt would remove. So massive addition of salt would remove bending by A-tracts. Really, this whole A-tract bending is really dictated by the persistence length. And I'm pretty sure, so if we would work at low pH, which we actually did, we did try to work at low pH. The problem is our protein COMK starts to form oligomers and everything is getting different. So I can't really comment on that, but I'm 100% convinced if we would measure at extremely high salt concentrations, the cooperativity would probably not work, or not work in the same manner. Yes. That's a good point.
Okay, we have a question from Rama Reddy Goluguri.
Hi, that's a great talk. I have a question regarding this Allostery. So, yeah, how much of this cooperativity is coming from just the spatial proximity of the binding sites?
Well, actually, you mean the spatial proximity of the 2 boxes, or within
yes,
of the 2 boxes?
Yeah.
Actually, yeah. I mean, this is the essential part of it. So if the boxes are closer together, then you have a much higher cooperativity when they are compared to when they are further apart. And sort of this is what I tried to explain with this worm-like chain model, where you can't get this cooperativity simply due to the polymeric features of DNA, under the assumption that the DNA is under some tension. And then you would expect this from simple physics-based principles. And
no, what I meant is when you have 2 binding sites nearby, so the effective concentration will increase for the other one, right? When one is bound.
Yeah. So you bind one protein or two proteins to one side, and binding to the other side is going to be increased if the boxes are closer to each other.
Yes.
What is the question? This is what you mean?
No, I'm saying you're saying that the cooperativity is transmitted through the DNA because of the tension and the DNA. So if you don't have that tension, you just have them spatially together, then it can also relate to some kind of cooperativity, right?
Absolutely, of course. Yeah, but I mean, these are so cooperativity effects due to protein-protein interactions are well known and well studied, and so I'm pretty sure, yes, you can have cooperativity effects due to physical contacts between protein molecules. I think, yeah, that's for sure.
Thank you.
Yeah, definitely.
Okay, we now have a question from your apartment mate here at Weizmann, who I'm sure is waiting for you to return. Phil.
Thanks, Gilad. Great talk, Hagen, as expected. My question concerns the physiological context. If you translate your findings on a rather truncated piece of DNA into full-length genomic DNA in the bacteria themselves, how do you think is this going to affect curvature, tension, cooperativity? Is it going to get stronger? Is it going to get, less obvious? How physiologically relevant are those findings?
Yeah, so making any prediction of what's going to happen in the cells, of course, means wildly speculating, but let me do that for a moment. So if we, based on what I showed, cooperativity is increasing the closer the boxes are. So if cooperativity is increasing, that means your function is getting steeper, which also means you will basically easier split subpopulation of cells that are competent and that are not competent. So the steeper the curve, the more clearer the splitting between competent and non-competent cells. And so if you would move the boxes very far away from each other, actually you would lose the ability to actually form stochastically such a competent phenotype, because you lose the ability of creating a threshold. So basically, you can get 20%, 80% of the cells being competent, which certainly is not certainly not of advantage. Now, to your question of how relevant these data are for the in vivo scenario, it's a very good question. We never checked it. My belief is, as you know, is that in vitro experiments have been proven for 50 years to be not totally wrong. And actually, if people, for example, study protein folding in cells or protein stability in cells, the numbers turn out to be, of course, slightly different, but the overall feature seems to be correct. So my big hope is, my belief is that what we found is not only a test tube artifact. But that's basically all I can tell you about it.
But you could make predictions based on these models, right? And engineer those strains.
In principle, yes, of course, that would be an option, right? I mean, you can put the spaces closer and further away from each other and make the spacer longer or shorter, and try to see whether you change the ratios of competent versus non-competent cells. And this is what I actually wanted to say towards the end of the talk. So maybe that's a pretty neat way of designing the behavior of genetic networks. In general, I mean, depending on how many other transcription factors are in this world that have these dual site promoters.
Okay, we have a question from Ronnie Granek.
Yeah, sorry, just a moment. Yeah, hello there. Hi, how are you? I hope my question is not is as smart as the other questions. What I get from you, I'm puzzled a little bit. So, because what I get from you is that actually the two binding proteins have preference to sit on the two sides of the DNA, because then the curvature is zero. Did I understand this correctly?
No, I don't think you understood this correctly, Ronnie. Okay. No, what I said, so the proteins are sitting on the same side, so they bind to the same side of the DNA cylinder. Yeah. They do this because the binding sites, in terms of base pair separation, are exactly 3 or respective 2 turns away from each other. That means whatever the binding site is, it will face the same side of the DNA cylinder. That's why the proteins bind to the same side. If I engineer a construct where the binding site is artificially placed on the other side of the cylinder, I find a factor 2 reduced cooperativity. This is actually the experimental finding. And so the idea is that it has to do with the energy that is required or that is released, I don't really know, when you bind proteins to this DNA and you change the curvature of the DNA. And so that, of course, will be different if you have a kink that faces this way and this way, or if you have this scenario. So that's all I'm saying. And so I have to admit, as I already mentioned to Jörg, that the theory is certain that we have available to sort of make rough predictions, at least qualitative predictions, is not ideal, because it assumes that it assumes an external tension that is applied to the DNA, and I don't see where the tension is applied to our DNA molecules. But nevertheless, the communication is apparently there. So all I can say is that either the tension is introduced already via those A-tract sequences, and so these curved DNA promoters are already under tension, which sort of would imply that they are not sort of the free energy minimum, but maybe they are. Or the theory gives a decent prediction, despite the fact that it's incorrect. So that could well be. Does it answer your question, Ronnie?
It clarifies something, because I was puzzled.
All right, okay.
It looks like, from your pictures, it looks like you claim there is preference where I could see only a frustration, and actually.
But there is clear preference so if you look at that, I mean, there is clear preference for having the binding sites sitting on the same side of the DNA cylinder compared to having them on opposite sides of the DNA cylinder. I mean, this is very clear, right? So the cooperativity values are much higher if the sites are facing the same side of the DNA cylinder. That's 2 turns, 3 turns, and that's 2.5 turns, basically. So this is for sure. There is an asymmetry between the two options.
Okay, thanks.
Yeah.
All right, it seems like there are no further questions. So I would like to thank you again for this terrific talk, Hagen. Thank you very much for jumping in at the last moment and for giving us such an exciting talk. And I hope to see all of you in 1 week in Nozomi Ando's talk. And thank you very much. And good night, good afternoon, good morning, wherever you are. Bye-bye.
Thanks so much, Gilad and Ben.
Thank you.
Goodbye.
Goodbye. Thanks, Hagen.